Good afternoon, all. I'm Dr. Pradeep Avle, Associate Professor.
आणि ती युट्यूबची पण लिंक ओपन करून देऊ मध्ये दिसतात
सर्वांना की त्यांचे आवाज ठेवा. The members for this online lecture in the memory of Dr. Daulat Rahe, sir. Today, today's session, I will request our Dean Academic Madam, Dr. Bruna Patel, to introduce the today's faculty, Dr. Anuradha Chogle. Dean Madam.
Very good afternoon. Thank you, Dr. Avle. Warm welcome, our Chair Professor, Dr. Anuradha Chogle. As part of our DS Hair lecture series, we have Madam with us who will be talking on role of molecular pathology in precision oncology. I'll just give you a brief introduction to Madam's achievements. Dr. Chogle has done PhD in biochemistry from BARC Mumbai University and postdoc from National Cancer Institute, USA, pharmacogenomics in pediatric oncology. She's an innovative, accomplished molecular biologist with 30 plus years experience in research and molecular diagnostics, with a focus on bench to bedside applications at the prestigious Tata Memorial Center, an advanced center for treatment, research and education in cancer, with 70,000 plus cancer patients registrations per year. She has received multiple awards. Just briefly run through some of them. Recipient of Gujarat Cancer Research Institute Oration Award, April 22. Pioneer in India for FISH technology in leukemia. Icon Best Project Award, funding for HDAC role in acute myeloid leukemia. Awarded CIPLA Kinetic Research Fellowship under Dr. M.F. Greaves, UK. And as chairperson, chief coordinator, has arranged various multidisciplinary preceptor programs for 50 plus cancer centers all over India for molecular oncology testing. As I mentioned, she is with us as chair professor in Maharashtra University of Health Sciences and is a chair professor of cancer genetics and genomics research at our Pune center. So once again, warm welcome to you, ma'am, and looking forward to listening to your talk on molecular pathology in precision oncology.
Thank you so much, Dr. Pradeep Avle and Dean Madam Runalin Patil, for a very, very warm introduction of myself. So today I'm going to talk on the role of molecular pathology in precision oncology. So it's just not the histopathology's role ends over there, but in this today's era of precision medicine, there is a big role of molecular pathologists to play in the precision oncology. So with the technical advances in the field of molecular genetics, it permits the precise genomic characterization of molecular tumors, as you can see, because the how the various signal transduction pathways are involved in the precision oncology. So it has not only improved our understanding of tumor biology, but also it has paved the way for molecular certified treatment strategies in routine clinical practice. So the molecular pathology analysis extends beyond the genomic characterization of molecular tumors by delivering insights into the behavior of a tumor and its response to various oncology treatment strategies. So in my today's talk, as you all know, the cancer is an uncontrolled abnormal growth of cells, but not related to any cancer particular cell of it. Like you can see, this is an abnormal cells which are or the immature cells in the leukemia which gets filtered into the blood. And in the solid tumors, it has the various cells which are involved, like epithelial cells, stromal cells, which get into malignancies. So this is the basis of our cancer, which is by definition, it is caused by an uncontrolled division of abnormal cells in a part of the body, and then it spreads to the other parts of the body through the blood and lymph systems. And it is just not one disease, but many diseases. As you all know, our body is totally of a heterogeneous cells, and that is why many, particularly in tumor, solid tumor, there are many cancer cells involved in it. So as you can see, it starts with the cell with the genetic mutation, then hyperplasia, then you can see the dysplasia happening, in situ cancer, and then you can see the invasive cancer. But how does cancer kill you? Really, does it kills you? Or do we have a strategic treatment in this today's era of precision medicine? So let us have I will touch upon the various types of cancers over here, and how histologically it has turned into the molecular characterization, and how the targeted therapies are involved in each type of cancer. So to begin with, it's traditionally chemotherapy works by killing cells that multiply quickly, whether they're normal or cancerous, as you all know. But however, the targeted therapy, it is a type of precision medicine. It works by stopping or slowing the growth or spread of cancer, and this happens on a cellular level. It can be at the extracellular level, or it can be at the intrinsic cellular level, or you can say call as the intracellular level. So the targeted therapies are designed to interfere with or target these molecules which are causing cancer, causing genes that create them. So the focus is on developing new targeted therapies that disrupt key drivers of cancer growth, but not all cancer types are currently identifiable as biomarkers. So the research enhances that treatment is led by biomarker testing, which extend patients' lives. So biomarkers can be used for several purposes. It can assess the individual risk of developing cancer, it can determine individual risk of cancer recurrence, and it can predict the likelihood that a given therapy will work for a specific patient. And also it is being used for monitoring the disease progression on to determine if a therapy is working or not. So we can do both to monitor the minimal residual disease and whether it is into the disease progression. So the biomarker testing has revolutionized the cancer treatment for many tumor types. Like you can see over here, all over the body, there are various cancer biomarkers are been detected, and these biological molecules produced by the body or tumor in a person with a cancer. So once identified, these biomarkers determines what is driving the tumor growth. So it helps to develop the treatment strategies, specifically target the tumor abnormality, and ultimately it improves the patient outcomes. So the biomarkers can be at the DNA, RNA, protein, or metabolic profiles, and which are specific to the tumor. So testing can include genomic testing to look at the DNA sequence, DNA or RNA test, or to look for the gene fusions, or test to measure RNA or protein levels. So as you all know, the central dogma of the molecular biology is DNA to pre-mRNA to mRNA to protein. That is, what are the steps involved? Transcription, splicing, translation, and the post-translational modification. So at what levels we can detect these molecular characteristics? At the genomic level, at the RNA, that is at the transcriptomic level, or at the proteomic level. So having said that, now there are various advanced technologies have come up which can be through which we can detect these various types of abnormal aberrations. So what are the at the proteomics? We can do the mass spectrophotometry. At the genomics and at the RNA level, we can do the gene chip method, or you can use a microarray image, or the latest is the NGS technology. So having said that, you can see over here that there is the graphic abstract which is published in the Cell. It says there are various cancer pathways, and the most recognized one are the 10 cancer pathways, which are like PIK3CA pathway, Hippo pathway, NRF2 pathways, Notch pathway, P53 pathways, RAS-RAF pathways, and as you can see, there are how many number of expressions are involved in it, in the each part of the body, and how there are almost more than 100 plus cancer drugs which are used as a targeted therapy by attacking the various signal pathways down the line are the RTK pathway, pathway, and the MAPK pathway is the main pathway, which is, and the mTOR pathway, which has not been mentioned here, but these are the various pathways where at the each genetic aberrations we are having the targeted therapies. So there is an integrated analysis of these genetic alterations in most of the 10 signaling pathways, and this article suggests that more than 9,000 tumors have been profiled by the TCGA. So cancer associated, there are all, it's other than the genomic alterations. You can see the cancer associated fibroblasts, that is mainly called as CAFS, are the predominant component of the tumor microenvironment and influence the cancer hallmarks. So the tumor microenvironment, where you get to see the epigenetic factors, are the main which stresses to form the abnormality or the mutations at the DNA and RNA level. So along with the malignant cells, you can get to see the heterogeneous tumor, that is the as I mentioned about the tumor microenvironment, and composites are the important parts of the tumor. So the TME interact with the malignant cells and contribute to the various hallmarks of the cancer. So, as we all know that the germline mutations and the somatic mutations are the main which plays an important role in forming of the cancer. Now, if you go to see the germline mutations, hardly 10% of the germline mutations are involved in happening of the cancer. To highlight them are the breast cancer, prostate cancer, then you can pancreatic cancer, and to some extent you can see and majority the breast cancer, ovarian cancer, and so on. So an alteration in DNA that occurs after conception is the one which takes place, and in the somatic mutation you can see they occur in any of the cells of the body except the germ cells, and therefore are not passed on to the children. So these alterations are nearly comprised of the 90% of the somatic mutation in cancer. So as you can see in this pictorial diagram, the variant present in the gamete and the variation, then thereafter the variation present in every cell in the body, including those of the germline. And the variants, these can be passed on to the offspring. Whereas and compared to the germline, the somatic mutations behave differently. They can be because of the various environment factors. Exactly today also the cause of the somatic mutations remains unknown. So these variants are absent in the gamete, but however, the variation can occur during the fetal development or any time after birth or in any cell of the body except those of the germline. So the variants are not passed on to the offspring when there is a particular somatic mutations takes place. So some of the better understood hereditary cancers I just mentioned, that the hereditary breast cancer, Cowden syndrome, familial adenomatous polyposis, multiple endocrine neoplasia, Li-Fraumeni syndrome, Lynch syndrome, retinoblastoma, von Hippel-Lindau syndrome. So these are the basic hereditary cancers are where the cancers are involved. So there are various mutations impact in the woman, in particularly when you're talking about the breast cancer, or you can talk about the ovarian cancer or the endometrial cancer as well. So you get to see the BRCA1 mutation, which can increase the chances of breast cancer and up to 81%, and ovarian cancer up to 54%. And it can be varied from the median age from nearly 30 to 80% at the age of 80. But the mutations in the BRCA1 and BRCA2 genes are rarely found in approximately 1 in 300 individuals in the general population and in 2% of the Ashkenazi Jewish individuals, but they significantly increase the chances of your cancer. Mutations in other genes are most but have less of an effect on the risk of developing hereditary cancer. As you can see over here, you can see the colorectal cancer, stomach, breast, melanoma, pancreatic, uterine, ovarian, where you get to see these germ-line mutations, where mainly other than the BRCA in the breast and ovarian, you get to see the APC in the colorectal cancer, CDH1 in the stomach, BRCA in the breast, CDKN2 in the melanoma. So these are the prominent germline mutations which have been identified in the germline cancers. What about the mutations which impacts the men? As I mentioned, same goes with the woman. Like in colorectal, you get to see the APC biomarker, stomach CDH1, melanoma CDKN2A, and in pancreatic it is the CDKN2A, whereas in the prostate it is a BRCA2, and in the very rare mutation, very rare cancer is of the male breast, where you get to see the BRCA2. So the APC mutation increases the chance of colorectal cancer up to 70 to 100% by age 80 if they are untreated. So what exactly are the mutational process? We get to see because of the DNA replication, genotoxins, DNA repair deficiency, DNA editing, you get to see these somatic mutations, under which you get to see the small mutations, and under which again they are comprised of single nucleotide variations, indels, that is insertions and deletions. Then you have the another somatic variant type is the structural variants, where you get to see the translocation, inversion, and the other types is the copy number variants, where you can have the copy numbers, which is very important in identifying the which type of cancer, whether it is, and also which is going to be a prognostic or a predictive marker in the various types in the cancer. So these I won't go much into the detail, but these are the steps: inversions, translocation, microdeletion, and large deletion, which comes under the small mutation. And in the structural mutation, you get to see some novel sequences, mobile element or pseudogene insertions, then tandem duplication, transposition, and novel sequence at the breakpoint region. And these are the laws of heterozygosity. It can be a loss or gain in this LOH copy number variants. This is the pictorial diagram again of the distribution of driver gene mutations in some tumor types. You can see at a glance, like there are, as you all know, there are various cell fate, cell survival, genome maintenance, cell proliferations, which are the different types which are involved in the cell regulation of the cycle. And you can see how the various types of mutations are involved in this under the cell fates, under the cell survival, and also under the genome maintenance. So this is a cancer genome landscape, which was published in the Science 29 in the month of in the year 2013. And you can see the identification of these druggable cancer drivers, nearly 73 types, and this is a very old paper, like in the of 2018 across TCGA data, but now it has definitely increased with the help on the basis of the research and how the genes have been validated, what are the prevalence of it, the basis of that, how the each loci of the chromosome, but the different chromosomes, how the mutations are involved, and how there are various types of targeted therapies available today. So this is the signal transduction pathways and targeted therapy in precision oncology. As I mentioned, there can be an extracellular or an intracellular pathway. And at the extracellular, patients are treated with the various molecular monoclonal antibodies. As you can see, the drug at the end, you can see the map, which indicates the nomenclature that this is a monoclonal antibody. Whereas in the intracellular level, you can see the drugs available with the nomenclature that it ends with a nib. That is, it is a small molecule, whereas this is a large molecule at the extracellular. And how the different therapies are available for various types of signal transduction pathways, and which ultimately affects the proliferation, survival, apoptosis, metastasis, and angiogenesis. So, but having said that, various types, you get confused where to target, what to target. So, this is what the different modes of targeting somatic mutations are available. So, at the I have shown you about the extracellular level, how at the with the various monoclonal antibodies you can treat the patient, but at the intrinsic level, that is at the intracellular level, how the cell behaves and what are where you all can target. So this is the ligand target therapy. Then you have the therapeutic antibodies. There is toxin at the extracellular level. Then you can get to see the tyrosine kinase inhibitors, which are at the intracellular signaling molecules. You get to see the apoptosis, agonesis, angiogenesis inhibitors. At the mRNA level also, now the drugs are available, which are called as the antisense. And of course, the immunotherapy, which is at the extracellular level. And you also get to see the metalloproteinase inhibitors. So in the immunotherapy, the popular one you look for is the PD-L1 and the PD-1 ligand molecules, which are being detected by the immunohistochemistry. So how targeted therapy works? So it doesn't kill like chemotherapy. It doesn't kills the cells, not only the cancerous but the normal cells. But its role is to block or turn off signals that tell cancer cells to grow and divide. It has also roles at the ATP binding site of the each DNA molecule. It prevents the cells from living longer than the normal, and and this is how at the after the patient responds to the targeted therapy for around 6 months or so, you also get to see the cancer cells are destroyed. So these are the successful stories of the targeted therapies. As you can see over here, BCR-ABL, which is a hallmark of the chronic myeloid leukemia, the patients have been treated with imatinib, and there have been successful stories. The patient the survival rate has beyond 10 years or so. Same thing with the HER2 receptor. When the patient is positive in the for the HER2 in breast cancer, you can treat the patient with the trastuzumab, and you have got the wonderful results of that. Similarly, coming to the lung adenocarcinoma, if you get to see the EML for ALK fusions, then you can treat the patient with the crizotinib. Now it's just not the ALK, but beyond that, we need get to see the red fusions. You get to see the ROS fusions, and we also get to see the MET fusions, which is or at the MET exon 14 skipping, what we call it as. And the same crizotinib has done the wonders, but also we have gone beyond that first line, second line generations, and we have got various third generation treatment also for these type of fusions, and the like ALK, RET, ROS, and NTRK fusion is there, then MET fusion is there, then the MET exon 14 skipping is there, and all this happens at the RNA level. Other than that, in melanoma, if this is the most prevalent BRAF V600E, and if the patients have been treated with the various type of signal transduction inhibitors. Similarly, with the lung adenocarcinoma, you get to see the EGFR, which is the hallmark of the disease. Like if EGFR is present, out of 4 exonic regions, exon 18, 19, 20, 21, you can treat the patient with the erlotinib, gefitinib, and the survival rate has definitely gone beyond 20%, which was once upon a time a very dreadful disease. So the genomic analysis is being incorporated into molecular pathology laboratories and into the clinical practice. So what all we need to see? The past and continuing molecular pathology methods, which involve the single mutation, single gene, few genes. And then under the genomic analysis, which are clinically useful, are the gene panels and the exome sequencing as well. So the genomic analysis, there is a lot, sky is the limit to do the research and look for the future potential to identifying the various signal transduction pathways which have been altered, which have still been uncovered or undiscovered. So the genomic analysis is the newest molecular pathology method which is happening now. So significance of molecular profiling in different cancer types, as I said, I'll be covering just touch upon the various types of cancers. So the NSA, non-small cell lung cancer, is the most common type of lung cancer and has been studied well. There is a lot of genomically diverse things which have been identified. Like as you all know, the predominant in the non-small cell is the adenocarcinoma and the squamous cell carcinoma, which comprises around 85%. But in this type also, adenocarcinoma is the most prominent one. But as you can see, there can in future are based on the driver actionable mutation. One can go with the WHO might come up with the various different type of classification based on this genomic profiling, as you can see over here. So in the driver mutation, the squamous, it are only the EGFR amplification is the main important alteration being found in the squamous cell carcinoma. So this was one of my paper which got published in the Annals of Oncology, where we did around 400 samples. Because today also we still depend upon the Caucasian population. The reference genomes is still being used as a Caucasian population. So this was one of the article of 1,000 patients done by the MSKCC, and they identified the various types of mutations involved. But here, when we did the Indian population study in the lung cancer, we found that other than these cancers, it is the percentage, as you can see, the percentage still varies with the with this known mutation. Still, there are some 43% of unknown mutations. But what we got identified was the FGFR3, that is the fibroblast growth factor receptor 3, which was highly identifiable or detectable in the Indian population, but it was absent in the 1,000 patients of the Caucasian population. So this was published in the Annals of Oncology, and this was a really hallmark for the Indian population study in the basically in the lung cancer. So a summarized landscape of lung cancer integrating predictive biomarkers, as you can see, from 1970 to 2000, these are the different milestones where small cell lung cancer versus the non-small cell lung cancer was identified by doing the various type of markers. As I said, it was the EGFR amplification versus the EGFR mutations, which were identified in these two types of cancer, adenocarcinoma and squamous cell carcinoma. So when we go ahead, you can see the EGFR and ALK were the mutations which were being identified precisely in the non-small cell lung cancer. And the other, beyond that, it was ALK, ROS, BRAF, CMET, HER2, NTRK fusions, RET fusions, which were identified when we came close to the 2020. And as per the NCCN guidelines, it has become mandatory to look for all these genes, particularly in the lung cancer. And not only now with the immunotherapy coming up, it is the PDL1, which is the platelet-derived growth factor ligand, which has been involved in the immunotherapy treatment. And this is the tumor mutation burden that is called as TMB, where we do with the vast 300 to 500 gene panel, we look for what are the different types of mutations are present. And if we calculate based on the how many mutations per MB are involved. So more the mutations are involved, it becomes compatible, or it becomes eligible to give a immunotherapy. So today, apart from these mutations, it is this PDL1 is done by the immunohistochemistry, and you can see that the tumor mutation burden by doing the large panel based on the next generation sequencing technology. So you can see how we have marched past chemotherapy, radiotherapy versus pembrolizumab, bevacizumab, and the TKIs. And now with the chemoradiotherapy, it is other than this chemotherapy, TKIs and immunotherapy is involved in the advanced lung cancer. So as mentioned earlier, these EGFR exonic region is comprised of nearly 29 exonic regions. But however, having said that, exon 18, 19, 20, and 21, the highest mutations have been involved. There are various alleles which are present in this mutation. So it is very, very important when you are doing an EGFR testing. It just not, we have to look for exon 19 and 21 or only 20. but we have to look for 18, 19, 20, 21, because you can see how many variations or insertions are involved in the exon 20. It is just not T790M, but other than that, you get to see the insertions, deletions in exon 20, and the most important one is the S768I mutation. So as we have matured towards the data, you are having more and more targeted therapies involved for each type of exonic region, 18, 19, 20, 21. So again, I will emphasize that all these 4 exonic regions has to be studied when the patient is a lung cancer, and in particularly with the non-small cell lung cancer. So this was my one of the established paper. Till 2013, we didn't have what was the overall frequency rate in lung cancer, but now this paper was published in 2013 in PLOS One, and with the 1,000 patient data, we could establish that the frequency rate of the EGFR is around 23%, which is much higher than the Caucasian population or to the Western population, but it was close to the Asian population. So with the care again, so with the various imatinib and the not imatinib, but the gefitinib and erlotinib and various targeted therapies, the survival rate definitely increased with this TKI therapy. But again, having said that, though they were exon 19 and patients were having these mutations like exon 21, but still there was not a good result happening, or the mortality rate was low in these patients. So what was the reason? So we studied along with the EGFR, we studied the KRAS mutation, and it was identified it is just not the EGFR, but the KRAS also plays an important role, whether it is mutant or whether it is wild type. Independent of that, we need to look for this KRAS mutation as well. And this was by another paper published in the British Journal of Cancer in the 2014. So I'm happy to share that a decade data of real world evidence of EGFR targeted therapy in non-small cell lung cancer has been published, and in this 10,000 in this 10 years we could publish around 9,000 plus data, and it was phenomenal statistic analysis which has been published. So around, it is a cohort of 9,053 lung cancer patients which were studied from 2011 to 2019. And we found an interesting finding that the baseline T790M mutation and along with that compound mutations were the only mutation observed co-occurring, while all other EGFR mutations were mutually exclusive. You can go through that paper. It's very interesting findings involved in that. So as I mentioned, the NCCN guidelines says that along with the EGFR, we need to study upfront the AL-CROS1, BRAF, PDL1. Then you need to look for other fusions as well, and at the baseline, that is as the first line. Why I am emphasizing this? Because it's a solid tumor, it's done on the FFP samples, and in the FFP samples, tissue is always an issue because there can be a small course where we need to extract the DNA very carefully, and if it goes negative, we cannot indicate that it's a negative, because if the tissue has been depleted, we have to be very careful. So it is the role of molecular pathologists to use the tissue very judiciously. We cannot do the testing one by one, but we have to have the panel of testing at the same time, and here it comes the role of the NGS. So this is again the histology in lung cancer. As you can see over here, from a morphologic diagnosis on H&E, the treatment was based upon. But now with the gene panel, we have passed upon with the various histological type, subtype, and has a broad molecular profiling. As you can see, as I mentioned earlier, is the RAS-RAF pathways, with the proliferation where it ends to the proliferation, cell survival. As I mentioned about the mTOR pathways, I again won't go much into the detail. So molecular subtyping of lung cancer has led to the approval and use of molecular targeted therapies, and this is how the NCCN guidelines is helping us to identify this. So now coming to the histological and morphological aspects of the progression of the oral carcinogenesis. So today the TGF, there is not much of a hallmark we can identify in the oral cancers, because oral cancer is again a subtype of head and neck cancer. So the TGF is there are some biomarkers which we can highlight in this oral cancers as well. So the transforming growth factor, that is the alpha TGF, is overexpressed in early oral carcinogenesis, and you can see that hyperplastic epithelium, and later by the inflammatory infiltrate, particularly the eosinophils surrounding the invading oral epithelium. So other than this, you also get to see the implications of viruses in oral cancer. So to highlight is the main is the human papilloma virus, which is also called as the HPV. It holds a prominent position. You get to see more than 77 types have been identified and characterized on the basis of differences in DNA sequences. So majority or the most prevalent one are the exon 2, 6, 11, 16, 18, 31, 33, 35 are the variant types of HPV, and they have been detected in oral epithelial dysplasia. So approximately nearly 33 to 50% of oral small squamous cell carcinoma are infected by HPV 16 and HPV 18. However, this HPV virus contain majority the two gene products, that is E6 and E7. So E6 is supposed to be considered as a high-risk HPV, which are capable of forming specific complexes with vital cell regulators, which binds to P53 and it induces the degradation. Coming E7 is as comparative with the E6, it has a low-risk HPV variants, and it interacts with the protein retinoblastoma, that is RB gene, and it blocks the downstream activity. So member of the herpes virus other than HPV, we also get to see the Epstein-Barr virus, that is the EBV virus, known as the EBV, and has been more frequently detected in the oral lesion, such as oral lichen planus and oral squamous cell carcinoma. So the histological differences, as you can, and the molecular aspects, I think this slide has got repeated. I will just shift to the next slide. But as you can see, the genes which are critically involved in the oral squamous cell carcinoma are the cyclin D1, P53, Ki61, retinoblastoma gene, EGFR signal transducers, and the activator of transcription 3 genes which are involved in the oral squamous cell carcinoma. Coming to the breast cancer, as you can see the various histological types over here, and you can see the medullary carcinoma, metablastic carcinoma, apocrine carcinoma, ductal carcinoma, mucinous carcinoma, cerebriform of carcinoma, tubular carcinoma, neuroendocrine carcinoma, classical lobular carcinoma, and pleomorphic lobular carcinoma. Now, in this era of this molecular, you can see how each biomarker has been identified by under the various histopathological classification. As you can see, luminal A, under luminal A, you get to see the estrogen, progesterone, HER2, ERPR positive, HER2 negative, and Ki67, which is at the low, which indicates the proliferation of the cell. So it has a very low proliferating cell, indicating it's a very bad prognosis. Coming under the luminal B, where HER2 is negative, but ERPR is positive, where there the Ki67 is very high. And then again under the HER2 positive, where ER is positive, PR is positive, and along with that HER2 is also positive. Then you get to see a very Ki67 mixed combination of low and high proliferation of the cell. But again, this is a very HER2 positive, all three positive hormones indicates that it can be very well treated with the hormone receptors treatment. So again, HER2 positive, but ERPR negative, you get to see the HER2 Ki67 high, and again in the TN, that is tumor nodes positivity, you get to see when there is a triple negative. What TN is indicates is the triple negative breast cancer, where all three are negative, and you get to see the frequency rate is around 10 to 20%. Having said that, this this triple negative breast cancer as of today is also very, very difficult to treat because there is no hormone receptors present on it. So as mentioned about the prognosis, luminal A type has a good prognosis, luminal B type has intermediate, HER2 positive has a poor, grade 3 present, and triple negative is, again as said, has a poor indicator. So we have different response to therapies present. So with the luminal A, there is an endocrine treatment, endocrine chemotherapy under this. You also, along with the endocrine chemo, you also get to see the targeted therapies. But again, targeted therapies along with the chemo are involved in this. But having telling about the triple negative breast cancer, only the what is available today is the chemotherapy. But these are the PARP inhibitors, which indicate if the patient is positive with the BRCA positive, then you can treat the patient with the PARP inhibitors like olaparib. So in the era of this modern medicine, only the morphological classification is not important, and the clinical pathological parameters which indicate the tumor size, lymph node involvement, metastasis are insufficient to predict the real behavior of the tumor pathophysiology. So as mentioned, there are various, we need to study the various molecular biomarkers involved in the cancer, in the breast cancer types. So more because each molecular subtype respond to therapy differently, also showing the variation in the prognosis. So coming to the ESMO clinical practice, luminal, we have the various guidelines. I won't go much into the detail about it, but today we need to follow this ESMO clinical guidelines for the treatment of the patient. Coming, as mentioned earlier, triple negative breast cancers. Now a lot of research is happening, and in search of new biomarkers, what we need to look for is the upregulation of RAS and MEK pathway, PI3K mutation, androgen receptor expression, somatic BRCA mutation, and that we need to study at the mRNA level. Coming to the ovarian cancer, yes, as you can see here, the subclassification: ovarian surface epithelium, fallopian tube epithelium, endometriosis, which is, and still there are some unknowns, and you get to see the various types: high-grade serous carcinoma, low-grade serous carcinoma, clear cell carcinoma, endometrial carcinoma, and mucinous carcinoma. So these are all the histopathology types, but do we have the molecular biomarkers to study in this? Yes, today we have, depending upon the which type of high grade or the low grade serous is present. You can see over here the various biomarkers are involved, like TP53, BRCA, NF1, RB1 gene, CDK12, homologous recombination repair gene, and this homologous repair genes is playing an important role in the treatment of this ovarian cancer. So under the low grade, you get to see the BRAF, KRAS, NRAS, ERBB2. Under the mucinous, you get to see the biomarkers like KRAS, HER2 amplification. Under the clear cell carcinoma, you get to see the ARID1A, PIK3CA, PTN, P10, CTNNB1, PPR2, alpha, and so on. Under the endometrioid, ARID1A, again PIK3CA, P10, and MMR deficiency of the patient. Coming to the non-epithelial sex cord stromal, you also get to see the granulosa, that is the FOXL2. Again, this is being well studied in today's literature, and others are including in the germline. So here in the germline, you get to see the if the BRCA is positive, you can treat the patient with the PARP inhibitor. Coming to the high grade and low grade gliomas. Now earlier, definitely there was no treatment available for the gliomas. It was only the chemotherapy and the surgery. But as you can see over here, under the low grade, you can see the various types of mutations present. K27M mutation. If there is no prognosis, we get to see the GTR mutation. If there is a K27 mutation, then it is considered as a high grade. As you can see, then IDH1 and 2 mutations are present, and the G34B mutation, with the G34B mutation, you get to see what all the protocols we need to look for: surgery, RT therapy, and the clinical trials, if at all any available. So this high grade are behaving very badly as of today, though we can see for the various RNA sequencing for that is NTRK, ALK, MET, ROS1. If with the 1% of these mutations or these fusions are present in this high grade, definitely we can treat the patient. But under the low grade, we can see the progression of BRAF mutation, BRAF fusion, FGFR fusion, or the mutations. And these are the molecular informed approach to the treatment, particularly in the high grade and low grade gliomas. I'll touch upon the classification of the ependymoma, which is a very rare type of cancer. And you can see under this, you get to see the ETMR 19q amplicons, and you get to under the posterior fossa, you get to, it has been classified as medulloblastoma. And in this, we can get to see the Wnt, CHH, group 3, group 4 subtypes. Again, there are SHH and so on. So we can see the ATRT, other embryonal tumors like ATRT, SHH, and ATRT MIC, ATRT TYR. So these are the sub-classifications of molecular subtypes under the ependymoma. So coming to the Gleason pattern, morphological, this is again a very rare type of cancer. So morphology and immunotype of this metastatic prostate cancer in a neck lymph node positive. So these are the Gleason tumors, prostate cancer. So we hardly 2 to 3% of the prostate cancers we get to see in compared with the lung cancer, breast cancer, head and neck. So the morphological, the tumor is composed of round cells with solid growth. Like you can see the tumor stain positive, nuclear stain positive, granular perinuclear pattern, PSMA, and the nuclear staining of androgen receptors. So coming to the, what are the biomarkers today are involved for the prostate cancer. So you can see the various type of cancer, prostatic intraepithelial neoplasia, we can get to see the CMEC, PCA3, MACR, ETS gene family activation via fusions, you can get to see the TMPRSS2 or other androgen receptor driven genes. So these are very well studied today, and various targeted therapies are coming up for this. So the prostate, which are under the intraepithelial neoplasia. So also you get to see the activation of beta-catenin bit loss of APC pathways over here. So having said that, if you have the loss of 8P, then you get to see the loss of P10, 13Q, RB1 gene, 5Q, CHD1, 16Q, and also you can get to see the gain of 8Q and 33Q location of this loci in the PIK3CA mutation. So also you get to see the inactivation of P53, RB1, and CDK12. So these are metastatic prostate cancer. They are still incurable, but if the castration and resistant mutation prostate cancer is present, still it becomes untreatable. So AR gene expression mutation gain under the B7 is a very, very important factor to treat them. So molecular subtypes of primary prostate cancers, as I showed you earlier, it can be ETS negative, it can be ETS positive. So under the ETS negative, you get to see the various types of biomarkers like IDH1, FOXA1, SPOP, FL1, ETB4, ETB1, and under the ETS positive, you get to see the ERG, which are the mutations present. So primary prostate cancers can be classified based on the molecular finding is the rearrangement of the ETS family, and you get to see the various transcription factors involved is the ERG, ETB1, ETB4, FL1, and which are ETS positive. So under the ETS positive prostate cancer, it can be classified by the specific ETS fusion gene member like ERG1, ERG, ETB1, ETB4, and so on. And importantly is the FOXA1 and the IDH1. So now I come to the prostate cancer I mentioned. Again, I won't go much into the detail. I've already mentioned which are the loci which are involved in this, what all chromosomal aberration you get to see in the prostate cancer. So 50% of both primary and advanced prostate cancers harbors ERG gene fusion or the ETS family gene rearrangement. So the primary prostate cancer without ERG rearrangement can be classified on the basis of SPINK1 overexpression, and this can be very well studied by doing the immunohistochemistry. Similarly, SPOP mutation and select SCNAs, which are often mutually exclusive. So nearly all advanced stage of prostate cancers have amplification or mutation of androgen receptors or abnormalities of AR pathway component. So you can see how we have progressed from the histomorphology to your molecular profiling. Coming to the hepatocellular carcinomas, yes, it is associated with diverse oncogenic pathways, and these are pinned under by different etiological factors, including virus infection, toxin exposure, or metabolic disorders. And the patient may display a varied genetic profile and may respond differently to the treatments involving the inhibition of targeted pathways. So the DNA, RNA mutations, copy number variations, chromatin structural changes, aberrant expression of non-coding RNAs, and epigenetic modifications are considered as biomarkers in the application of molecular profiling targeted therapies. So again, this is a hepatocellular carcinoma. As I've already mentioned, in the paucity of time, I won't go much into the details about this. So you can see over here how the basis of the molecular aberrations involved in the RAS-RAF pathways, apoptosis, cell survival, migration, invasion, and proliferation, which encodes the proliferation and angiogenesis. We have the different types of treatment like lenvatinib and sorafenib, and so many drugs are involved in the targeted therapy. So, sorry for the slide. So there has been, as you have seen, that there has been a paradigm shift in cancer diagnosis from histopathology to molecular. But having said that, has histology lost over biology? Yes and no, because we have moved from just from the microscopy to a important technologies based on the genomic and profiling and the transcriptome profiling and the proteomic profiling using the high technology like microarray and NGS and so on. So the 3 decades ago, diagnostic histopathology has an endpoint, but what one could do with the classical morphology. But having said that, will morphology disappear? No, because will cell and tissue samples be subjected immediately to sophisticated molecular analysis, foregoing on the more global perception of the condition at a microscopic level? Yes. Will body fluids like liquid biopsy replace cells and tissues as substrate? Yes, we can. But however, the liquid biopsy cannot tell the origin of the tumor. But a lot of research, and now also into the clinic, we are looking for the cell-free DNA for the profiling of the each cancer disease. So who can tell? The art of prediction is difficult, notoriously so when it comes to the future. But this is a good moment to reflect on where we stand today and what the near future might look like. So, although histomorphologically has made significant advances, unlike understanding of cancer etiology, classification of pathogenesis, it is sometimes complicated by morphologic ambiguities and other shortcomings that necessitates the development of ancillary tests to complement its diagnostic value. So, a new approach to cancer patient management consists of targeting specific molecules or gene mutations in the cancer genome by inhibitory therapy. So just I will skip this slide because okay, coming to the so what is the role of the histopathologist when there is a tumor? So assessment of the tumor topography, cellular morphology, classification, nomenclature of this, it adds values to the patient's management by identifying certain prognostic indicators such as lymphovascular invasion, infiltration of adjacent organs, necrosis, and mitotic rate. So few but important advances in the field is the immunohistochemistry, immunocytochemistry, digital pathology, and so on. So what is the major difference between the histopath and the molecular profiling? Histopath, the task is for the tumor classification, grading, and staging, whereas the genetic characterization of tumors has gained a tremendous speed during the last 10 years by the development of the NGS sequencing. Histopathology has improved knowledge about genetic aberration, which has led to the molecular development of subclassification of the tumor, whereas at present, a single gene analysis with mutation-specific PCR and Sanger predominates in the diagnostic molecular pathology. Our gene panel sequencing with NGS has increased rapidly. So the pathologists of the future need to combine the morphological methods with practical and theoretical knowledge in genetics, cell biology, biochemistry, and bioinformatics. And but also the NGS, that is molecular scientists, they need to analyze the NGS data is very challenging. So also we need, the scientist has to understand what is the pathology or the etiology of the each tumor, and pathology depends, and departments have to recruit and integrate bioinformaticians to master the increasing demand for genetic tumor profiling and correlate with the histological findings, as I said. So I'm going to skip this slide. Yeah. So what is the connection of the histopathologist to molecular pathologists? What is the journey today? So examination and dissection of the surgical resection specimens to select the most appropriate sample for the microscopic slides, and the microscopic examination of the tissue, and the construction of the clinical report. So what is the connection of the molecular pathologists? These molecular pathologists have to understand the genomic and the genetic knowledge about each transcriptional pathways aberration. They are now required not only provide the diagnosis, but also study the precise molecular characterization of each individual cancer case and play a significant role in the treatment of planning of the patient. So molecular pathologist is supposed to be a surgical pathologist, and also he has to understand the molecular biology of each cancer disease. So, yeah, so stay informed on treatment information related to molecular analysis, helps ensure the biological activity of the tissue samples, provide clear, concise, and comprehensive reports, add a few more data points to your specimen, processing protocols to ensure the compliance of the routine molecular oncology testing. Once molecular tests have been ordered, refresh yourself on the basics so that you know the best sample to provide. Check the biomarker results, correlate with the clinical profile for each patient. And if you have an in-house molecular laboratory, nothing like it, because it controls the quality, it controls the turnaround time. So it is best to have your own molecular laboratory in-house instead of outsourcing the samples to the various labs. So stay abreast of new guidelines for testing, attend the tumor boards or multidisciplinary treatment planning discussions, be available as a diagnostic consultant, and actively participate in these discussions and share information on how molecular testing can be used to inform clinical diagnosis and the treatment decisions. So these are the advancements in the pathology, as you get to see, other than the microscopy or the tissue processing. Now we have got, earlier it was just the manual method. Now we got the automated staining for IHC staining as well. Under the digital histology facility, the workflow, now there is an image processing, and where we can use the stain slides, and also the whole slide imaging has been done. But the artificial intelligence has a next step forward for this precision pathology. So we need to look for the artificial intelligence where it gives, emphasizes, focuses on the decision support tool, outcome prediction, predict molecular profile. And so definitely the future is the artificial intelligence in the molecular pathology as well. So having said that, these are the how we have marched past from the histomorphology to the molecular diagnosis. As you can see over here now, based on the DNA, RNA extraction, of identifying with the various technologies like Sanger sequencing, RT-PCR, FISH, immunohistochemistry, and now with the next generation sequencing, we get to know the SNPs, CNVs, and whole insertion, deletions, epigenetic profile, everything can be done under the one umbrella of next generation sequencing. So this is how the advanced techniques from histopathology to molecular pathology. We had IHC, FISH, PCR, Sanger, NGS-based hotspot testing, hybrid capture, like FISH methodology, and now the whole exome sequencing and the whole genome sequencing as well. And now with the whole, we can say the transcriptome studies, it has impacted well, increases as methods becomes more rapid and less costly, and ultimately are being used to generate comprehensive genome profiling. So these are the various methods like Sanger sequencing. I won't go much into the detail, where only there is a tumor limitations. At least we need to have the 20% of the tumor to do the Sanger sequencing to rule out the false negative results. Real-time PCR, it is highly specific and sensitive, but again, it has limitations. It can detect only a single gene at a time. But now, again, we are getting some new markers coming up, small panel coming up of the lung cancer, like 9-gene panel. We can do it on the real-time PCR as well. Digital PCR, yes, it helps in monitoring the residual disease. It because each molecule is been partitioned and each droplet prepared is again exposed for the PCR amplification. That is why it becomes very highly sensitive and specific methodology, and that is why it helps in monitoring the disease. Again, as I said, coming to the Sanger sequencing, it is next generation sequencing. It is a sensitive method, expensive and time intensive. However, now with the maturation, with the mature data and with the mature technology, we are coming down with the low cost. Turnaround time is also being less, but it sequences millions of molecules in parallel and do not need prior knowledge of what you are sequencing. But of course, if, other than the wet lab, it has somatic clinical bioinformatic pipelines are involved. So once the raw data is generated, we get to see the FASTQ files, and these FASTQ files are then filtered, trimmed, because we have the barcodes of each sample. We need to remove these adapters. Then there is an alignment. Then there is an aligned read conversion that is called as SAM to BAM. That is significantly they have aligned to the various types of mutation. So VCF, again, then once this is done, we get to see the VCF, that is the variant concordance, variant filtration, variant detection, where we get to see the single nucleotide polymorphism indels, and the indel realignment is done, and with the variant annotation, variant prioritization, variant visualization, and then the clinical report is given. So we have to be very careful. We need to have a good bioinformatician to do the analysis of this, and the various tools which are involved are the VWAVM, SAM, SMT tools, GATK, BCF tools, and Varium MAT and Webinar. These are the in-house tools which can be used for the bioinformatician to generate the clinical reports. So there are various cancer databases, cancer anatomy projects, where we need to correlate with the whatever the mutations we have been identified. So there are like WWNCBI, we have the cancer.sanger, then we have OncoKB, we have intogen.org, then we have My Cancer Genome, then we have the CLIMVAR, which I forgot to mention over here, which is most prevalently used for the raw data analysis. So where you get to see the type of genomic alteration detected by NGS, that you can look for the point mutations, you can look for the indels, you can look for the homozygous deletion, hemizygous deletion, gain, translocation breakpoint, and whether we can also identify whether it is highly pathogenic or it is non-pathogenic or likely to be pathogenic or it is variant of unknown significance. So as oncology testing expands, laboratories are challenged with finding highly characterized, standardized, and reproducible quality control materials to support the validation or verification of these new mutations identified. We need to correlate with the patient results, or we do need to correlate with their clinical findings as well. So how can molecular tests help with the cancer? What is the next generation sequencing, and how accurate is molecular testing? So in this era of precision medicine, we need to have an equally powerful quality management system, that is the QMS, to assure consistent, reliable data at any scale. So the quality of the test result is linked to a number of factors. The GCLP for molecular-based tests, potential of molecular genetics initially recognized by oncohematologists to detect specific chromosomal translocations, then emergence of practical applications of molecular oncology, and NGS as of today is a game changer, comprehensive description of germline DNA, analysis of somatic mutation, and so on and so forth, like RNA profiles. But however, when you are into the clinic, this can be accepted in the research lab, but if you are going to use NGS in the clinic, then this chaos, which is you can see over here, is definitely not acceptable, because you don't know, are you looking for a right technology, or there is a contamination we cannot rule out over here. So this is the very scary part. Whoever is in charge has to have the hold on the quality control. That is what it indicates. So total laboratory process in NGS, again, you need to have the assay validation. There has to be a validation for at the pre-analytical, analytical, and the post-analytical levels. We need to consider for the special consideration that the clinical validity, utility of nucleic acid test, providing clinical interpretation of the test results, staffing and technical expertise. This is a very, very important factor. You just cannot keep on roll pay who are the technical staff who has not been exposed to the NGS. There has to be a regulatory oversight and the best practice considerations as well. So, outline establishing quality in the molecular laboratory is the most essential role, and the molecular pathologist has to look into these points very well. So, practicing continuous QA, we need to participate in the ICP program or in the proficiency testing as well. So, we need to follow the CAP guidelines. So, the special consideration for molecular diagnosis laboratory, as I mentioned earlier. So this was one of my paper again, which got published in MDPI last year, how the comprehensive development and implementation of good laboratory practice for NGS is very, very essential. And this was done around 144 samples. We use the targeted panels on solid tumor using the FFP tissues. And you can very well look upon this paper, how a decision tree is made to perform the NGS on the targeted panels on solid tumors. So take home message, this is my last slide. Histology cannot be ruled out. However, it coexists and complements with the molecular testing, which can provide comprehensive cancer diagnosis. In the era of precision oncology, genome profiling is the grammar. However, the histopathologists dire need to look beyond microscopy. So the future development and challenges in molecular pathology has to be considered. Molecular tumor board meeting should be an integrated part of the reporting. And in this MTB, we need to have a panel of medical oncologists, surgical oncologists. We need to have a molecular pathologist and also the molecular scientist who has performed the NGS and who can very well explain the mutations present and how you can interpret for the targeted therapies. So to acquire high throughput technology in molecular diagnostic laboratory is mandatory for the GCLP, good laboratory practice. So improve constantly and forever the system of production and service to improve quality and productivity, and thus constantly decreases the cost. This was mentioned by the Edward Deming. And with this, I stop over here. Thank you for the patient hearing, and I'm ready to take the questions. I'm happy to announce that Gene Health Laboratory has organized a preceptorship oncology workshop for a 2.5 day hands-on workshop with the 10 candidates on cancer biomarkers. So also you will get to hear some brainstorming lectures by the medical oncologists, interventional radiologists, molecular pathologists, and the scientists who are involved in this technology. The dates are 8 to 10th November. Registration fees are around 5,000. Please visit the MHS site, or for details, you can contact Dr. Anuradha Chaugale, that is me, or Dr. Brigadier Dr. Sen, who is the HOD of the Gene Health Laboratory. And thank you so much. Hello.
Thank you very much, madam, for your wonderful presentation.
I'll stop sharing my slides.
Yeah, please. I request Dean madam to speak few words.
Yes, ma'am. Actually, the dates of that workshop, you mentioned, did it take place between 8 to 10th November or?
Sorry, it's not November. The date, I just forgot to change the date. It is 8th November, 8 to 10th December.
Okay.
Yeah.
And
so.
Yeah, I mean, it definitely added to my knowledge.
Hear you, your voice is cracking.
I said, yes, today I very patiently heard each and every word that you were talking and really added a lot to my knowledge where precision oncology is concerned. right from biomarkers to the various pathways to germline cancer to somatic mutations to most importantly how the diagnosis as well as the treatment of cancer has changed by leaps and bounds and how we all need to keep pace with the changes that are taking place. And it's good work that you're doing at our genetic lab also, especially your work in oral with this lung cancer. That's really appreciable.
Thank you so much, ma'am. Any questions? I'm happy to take.
Madam, questions will not come here because we have restricted the entry. So,
Okay.
so I would like to thank our Dean Madam and Dr. Chokule Madam for a wonderful presentation. Thank you very much, all of you. Thank you.